Tēnā koutou katoa, he tino mihi ki a koutou. Kua tae mai nei, ki tēnei hui rorohiko o te ahi nei. Ko te mea tuatahi, ko te karakia tīmatatanga o te wā nei. Nō reira, e whakawhetai ana mō tēnei rā, e whakamaumahara ki ngā taonga tuku iho, ngā maunga, ngā awa, ngā moana, ngā rākau, ngā manu, ngā koiora tuku iho. Hei tiritiri mō tātou katoa, o tātou wairua, hei hanga tangata kōtahi, kia mahi tahi ai mō te ao hou tino pairawa. Nō reira, tūturu whakamau kia tīnā. Haumi e hui e, tāki e. Kia ora koutou.
Me is Sean there for that karakia, and I've just noticed actually that Sean and I have one of the same posters in the background of our video, so that's great. Yeah, good afternoon everyone. No mai haere mai, welcome back, or welcome if this is the first session you're attending today. My name is Taylor and I am the MC for today's proceedings of this awesome day 4 of what's been an amazing conference so far and will continue to be. Just before I kick off into anything else, a few housekeeping things. Many of you probably all aware of this stuff by now, but I'll remind everyone anyway. If you experience any technical difficulties, there's teams here that can help you make sure you've got all that sorted and that you can enjoy the conference. There's easier to watch streams with lower latency that can help if you've got any network issues. There's closed captions on all of the talks, and all of the talks can also be watched for 3 months afterwards as well. So you can catch up on any of the kōrero that you might have missed because you were out or anything like that. In any of our breaks, make sure you use the meeting hubs to catch up with people you might want to chat with, but also go check out the exhibitions of all the sponsors and supporters of the conference as well. And of course, make sure you're interacting with the platform. There's lots of ways you can earn points and win prizes, and that's a really cool feature. And while we're watching all the kōrero today, make sure you chuck in your questions and comments into the live Q&A and discussion forum. It's really good if you're going to chuck a question in, chuck it in before the talk ends so that we can see it and that we can ask the speaker and get a good answer for you. You're just going to have to bear with us a little bit in this session as we've had some changes around due to some speakers being absent. Very sad. Aitai will not be speaking today, and so we'll be moving on straight to our next session, and I'm going to introduce Simon Palmer, who's going to be the chair of that session. So many of you already have seen Simon have a role in this conference so far, but Simon is a researcher based at Te Kawa o Te Maui, School of Māori Studies at Te Hereanga Waka Victoria University in Wellington. He works as part of the Novels, Tools and Strategies theme in Bioheritage Challenge and also co-leads Ngā Pikarere, the Early Career Network. So I'll hand over to Simon to lead us into this next session.
Tēnā koe Taylor and tēnā koe Sean. Kei te mihi au kia koe mō te karakia. Yeah, awesome to be back and looking forward to this session. So I'm just going to introduce the host and pathogen and environment video that's coming up. The host, pathogen and environment team is focusing on the disease triangle: host susceptibility, the pathogen, and the right environment for disease expression. They are investigating the role environmental factors play on Cody dieback and myrtle rust disease expression and severity, as well as researching the pathogen genomes. The knowledge they gain will contribute to improving surveillance, control, management, and conservation efforts, and they hope to discover new ways to mediate these diseases. Kia ora.
Ko manaia te maunga, ko whāngarei te ringa parawa te moana, ko poki kauri te awa, ko takahiwai te whenua, ko takahiwai te marae, ko patuhara keke te hapu, ko Ngāti Wai o rawa ko Ngāpui oku iwi, ko Julianne Cheetham tōku ingoa. Our team is the Patuhara Kihei Tiwi Trust Board has a tile unit, and they've been sort of functioning for several years now, and they've been undertaking quite a lot of work up at Pukekohe, which is the whenua up on the ranges, Takahiwai ranges behind our kāinga, behind our marae. It's a really significant place for us because our rangiora pā is up there and other important pā as well, and the land is regenerating kauri forests. There's thousands and thousands of kauri up there. It's some pretty healthy bush up there. You would have heard in my pepeha I mentioned Pukekohe Te Awa. Pukekohe is a water body up the top there in the ranges, which actually is the source of our Takahiwai awa, which flows down into the harbour. It's a really central part of our identity, and one of the things I've been always sort of saying since being involved in the kauri kaupapa with this disease is that, you know, it's a threat to our identity. You know, when we say our pepeha, when we talk about these significant places, how can you have a Pukekohe that doesn't have kauri on it? It's a real, you know, it's a real risk to our narrative, our kōrero, all those things that we pass on to our tamariki and mokopuna. From the beginning, we've been really interested in finding out all we can about this disease, this sickness, mate of our kauri, and how we can protect this sort of last place. And we know that we've got this disease in other parts of Arohe and the Waipu end, over the back of Ruakaka on the Abakukanui side, but we were always pretty sure that Pukekohe was clear or clean until a couple of years ago when we did have some samples in 2019 that came back. Just a small handful of around a couple of trees, and then we were really, really concerned because we thought we might have a new infection in there. But then our more recent testing hasn't been able to re-isolate the PA. So we're kind of in a situation now where we may or may not have kauri dieback up in the Pukekohe blocks, and we really, really want to know one way or the other, because that will affect, you know, how we manage and make decisions going forward up there with all of our kaitiaki practices and things, not only that the tile unit does, but also that other whānau, you know, are doing up there. So you will have heard a lot about the disease triangle, and that is basically what this research is doing. It's looking at the host, kauri rākau, and the pathogen, Phytophthora agathodicida, and also the environment around it. So it's a whole of ngāhere approach, which is really coalesces really, really well with our Te Ao Māori thinking. We don't want to just focus on what the disease is doing or unhealthy trees. We want to look at what makes the ngāhere healthy or unhealthy, and how is the pathogen moving through our ngāhere or not.
So we're trying to map at the landscape scale areas that are more likely to show sign of decline in the canopy for the kauris, or symptoms from it to rust. And the way we do that, we're currently working with Naris and their team to identify some environmental variables that favor the expression of the disease. So whether it's areas that are downslope a track, for example, or downslope trapping lines, area that are a bit more wet than others. We use like that different factors that we try to include in the model, and then the modeling helps you identify those areas that are more at risk than others.
So it's looking at what the soil's like. Are there other types of Phytophthora present that might be impacting the trees as well? And what else is happening in the catchment water-wise? What's the topography like? Is there a lot of pests and weed and just other health factors that are impacting the health of that ngahere and causing that disease to get a hold and sort of start spreading.
So I think the idea behind all this is once you have this vision, you could, based on this, what we call expert knowledge, we could say in that landscape, this area is most likely to be infected than this one. So this is maybe where we want to concentrate our efforts to identify the disease front and maybe protect those areas that hasn't been infected yet.
You know, it's quite difficult because you don't always have symptomatic trees. You don't know exactly where the pathogen is in the soil and where it's moving, you know, down a catchment or in what part of the ngahere and how fast is it moving. And those are the kind of things you need to know if you want to make management decisions. And that's also the kind of thing that mana whenua really want to know when they're trying to look at what they can do in their own forests to protect kauri.
So we've been involved with Pathogen, the team at Pathogen. We had meetings and trying to identify the specific trees that they would go and sample within the forest. So there was a process there where initially there was some identification of emergent kauri trees from the canopy, and that's done through aerial flights. And then once we had all the data on the trees that were in the Takahiwai forest, we had to, obviously not every tree can go and be sampled. It would be great, but there is not the resources. So we randomly selected a group of those trees in conjunction with Julianne and her team.
When we were doing the site selection for this with Ngāri and the team, and also some of Waitangi and Dean's team, we were looking at, there was sort of, we were doing randomised sampling, but then we were doing selective sampling over that. So we were saying, well, what's on our rangiora pā do we want to look at? Because we looked at our rangatira rākau there in that area before, so we know we want to go back there. Are there any sites that have been randomly selected but fall into those areas, because those are ones we want to focus on? Or we might look at different whānau blocks and think it's all well and good doing, remote sensing and getting the data, but we actually know from on the ground, well, it's going to be really nigh on impossible to get up to the back of that block because of the terrain, or we know that we have whānau or neighbours that are not going to want us to go up through their property. So applying all of that local knowledge has been part of the process as well.
So we went through that process with them and created some maps that they could then go into the field and find the tree and organize their field day, I guess the day in the field, and which group of trees they could go and sample. And from those trees they did some visual assessments and some soil sample. A group of them did soil sample, and the soil sample goes back to the lab with Ngāri. All the data that is collected using this survey form, but whatever gets collected, then as soon as the field day is finished and you've collected that data, the data goes back to a central, data server, which we're not yet using the IIP because it's still in development, but ideally the process or the workflow is going to be that any field data collected is going to be stored straight into the IIP.
That's why our team, our Taio team, are really wanting to be at the forefront of the Research Area 3 mahi. We've had some training from BioSense. We've got new gears. So we've got, new tablets, new GPSs, all the gear to make sure that we can do sampling and surveying that's consistent with the approach that's been taken in the Waitakares. So the methodology that's being used is really consistent, and it's going to be applied in Te Roroa and Waipawa, Ngāti Rehia, Ngāti Rua, Te Uri Hau and Tauranga Moana. So there's quite a few mana whenua that will be utilising this approach. I think our young fellas have been really good in tweaking the survey methodology, what works on the form, and practice in the ngahere, which is going to be put, I think, is going to put all the other BMAs that are going to be starting this work very soon in good stead, because we've almost been like I guess the guinea pigs or the pilot, sort of saying what's doable, what's doable on the new tablets, how does it all sort of come together, how many trees can you sort of sample in a day, and we're sort of working out what the work what the work plan is and how much capacity and capability is needed. But we have been here for a long time. Our marae at Takahiwa is the only marae between Nguru and Omaha marae. And we have been really active in an environmental mahi since forever. What's good about the mahi up at Pukekohe, there's a mamae sitting under it, so it's about how do we get this place back, but at the same time it is kind of more proactive, positive work in terms of our tai tamariki and our team going up there and being able to, have a look at what's in the waterways. We've had 2 wānanga up there where we've had our weavers from Patuharakeke and other whanaunga from around different rohe come to do kuta workshops and things like that up there. So we're trying to revitalise those practices, other rongoa, rāranga, those kind of activities. And this Kauri dieback, Kauri epidemiology research really feeds into this whole interconnected sort of kaupapa. How do we reconnect with this place, and what kind of practices do we want to do up here in the ngahere? Everyone's really into it, like not just our young fellas that are going up and doing the mahi. We take a lot of our kura kids up there and show them what we're doing up there. But it's also our koros and nannies and that are really keen too, because they don't get to go up there that much, and they remember the stories about what it was like up there. There used to be waterfalls up there and things like that are gone now because of the dam. People used to go up there to get pigeons and all of those kind of things. So we're trying to collect all that traditional kōrero as well as part of this and other projects we're doing. So it all ties into, real kaitiakitanga, what we're trying to revitalise and achieve up there.
Fantastic. That was another beautifully shot and informative video from the host pathogen and environment theme. We're now going to move to a presentation from Julia Soewarto. Julia is a forest pathologist at Scion in Rotorua and has 8 years research experience working on myrtle rust in both New Caledonia and New Zealand. Her research is focused on biosecurity, disease management, surveillance, and plant resistance, with a specific emphasis on indigenous tree species. Over to you, Julia.
Thank you. I have a bit of feedback. Can you confirm that you see my screen? Good, thank you. So thank you very much for this opportunity to present on Nyataras today. So this is going to be a short presentation on some of the Nyataras work that we've been doing since 2018. And the idea was to assess and monitor the impact of the exotic pathogen Nyataras on New Zealand native Nyataceae. So lots of people are involved in this work. So people from Scion and also colleagues from Plant and Food Research. And this research is funded by Narako Taketake under the Host Pathogen and Environment team. So a bit of introduction about myrtle rust. So myrtle rust is a disease caused by a rust fungus called Ostropocynapsidae. Often the symptoms are these yellow spores that are developing on the myrtle plant. It is native to South America. It is a wind-dispersed fungus that was introduced in New Zealand in 2017. It was first detected in Raoul Island in the Kermadec in March, and shortly afterwards it was found in a nursery in Kerikeri and then in Taranaki, and then it spread from there. So the disease is now found widely across the North Island and in some areas in the South Island, including a recent report from the Chatham Islands. So myrtle rust is here to stay. It cannot be eradicated from New Zealand. So following the initial incursion response, which was trying to remove infected plants, we quickly moved the focus of our science to manage the disease in the longer term. And part of this longer term management is to understand how much impact this disease might have on our native myrtle. So myrtle rust only infects plants belonging to the myrtle family, which include many native and exotic species in New Zealand that have a cultural significance or that are ecologically important or that are grown for commercial purposes. So currently, there are 32 myrtle species that can be infected by myrtle rust in New Zealand. These include 18 native New Zealand myrtaceae, which represent more than half of all the known native myrtle in New Zealand. The most common hosts are species from the Lophomertus genus, so Ramalama and Routu. Metrosideros species, and especially Puhutukawa, are very susceptible to myrtle rust, as well as Syzygium species like swamp mire. So back in 2018, we wanted to look at the impact of myrtle rust on natural population of the very susceptible species of the Lophomertus genus. So we have established a network of monitoring transect across a range of different locations in the North Island of New Zealand. And we looked at the impact of myrtle rust on seedling recruitment, plant growth, and flowering. So in our most infected sites, it's been a full season that myrtle rust has been hammering the Lophomertus trees. And we have seen a significant defoliation and decline in tree health. We have also reported an interruption of flowering for the most infected trees. So after all these years, we haven't reported tree death yet from these monitoring sites, but we know that death of mature Lophomertus trees is happening in other regions, such as the East Cape, for example. So myrtle rust affects all stages of the Lophomertus life, including flower birds, flowers, and fruits. It prevents plants from producing seeds and so prevents it from producing offspring to replace the dying parent. Another very worrying news for Lophomertus population is that we haven't seen any evidence of new seedlings or recruitment since December 2020 in our most infected sites. So probably the most important message from this talk is that it is very likely that the progressive decline of Lophomertus population will lead to extinction of this species, and the loss of Lophomertus species could be associated with broader ecological impact and flow-on effect on birds, insects, other plants, and microorganisms. And unfortunately, Lophomertus species are not the only species threatened by myrtle rust in New Zealand. We are now seeing similar impact on Syzygium mire, which is the only Syzygium species, native Syzygium species in New Zealand. So is there a future for Lophometer species where to save this species from extinction, we have to rely on conservation strategy. And so recovering action may look like trying to identify resistance within populations of Lophometers and then collecting seeds from these resistant individuals and redistributing them back into the environment as part of a rewilding plan or regeneration program. So here you have an example of a Lophometus tree that is very susceptible to myrtle rust and had its growth very much impacted by the disease. And on the right side, you have an example of a tolerant or resistant tree that is showing a good growth. So that's all for me. Please feel free to ask a question or get in touch if you would like more information.
Kia ora. Awesome. Thanks for that presentation, Julia. I'd now like to move on and introduce Rosie Bradshaw, who is a professor in genetics at Massey University in Palmerston North. Her research is mainly focused on understanding forest pathogens using the tools of molecular biology and genomics. She works with a range of pathogenic species, including the fungus that causes Cody dieback. Over to you, Rosie.
Opportunity to talk about our new genome sequence for the Cody dieback pathogen. So in this short presentation, firstly, I'm going to talk about why genome sequences are important. Then I'll talk about why we need a new genome sequence for this pathogen, and then I'll tell you how far we've got so far. So genome sequences are very, very useful, and I want to just illustrate this first by something that unfortunately has become only too common around the world and well known to most people, which is the virus that causes COVID-19. So the genome sequence for this virus is relatively very, very small, has only a small number of genes, but it's been absolutely essential for developing the diagnostic tests that you'll be well aware of and probably have used, for detecting new variants of the virus and where they originate from, and also for understanding the pathogen, the biology of the pathogen at a molecular level. So by this understanding, it's been possible to develop control methods. And in the case of this virus, as you'll realize, vaccines have been produced, and that's based on the knowledge, for example, of the spike protein, which is that yellow looking molecule which is sticking out of the virus in that picture. So the Phytophthora agathodista genome, in contrast, is much larger. It's almost 2,000 times as big, and it has far, far more genes. So all of these uses that are listed here and more apply, and you heard a bit about diagnostic testing in the video that you just saw. But understanding the pathogen is far more complex, as you might imagine, with so many genes. So we really tick the box here of the crazy and ambitious project to try and understand the pathogen at the molecular level and with such a large genome. But ultimately, by understanding the pathogen at this level, we hope that eventually there'll be new methods of disease control. So why do we need a new Phytophthora agathodista genome sequence? So a genome sequence for this pathogen was done at Scion a few years ago, or quite some years ago. That was using the best technology that was available at the time, the so-called short read sequencing technology. And this is kind of like having millions of very, very tiny jigsaw puzzle pieces and trying to fit together the jigsaw puzzles that are the individual chromosomes that carry all the genes for an organism. So they did a good job. They got down from millions to 3,689, but still many genes missing in the gaps, as you can see in that little picture bottom left. And some genes were cut in half. And really, to get a proper picture of the biology of the pathogen at a molecular level, we want to make sure we're not missing any important genes. So we really want to have a genome sequence with all the genes. So sequencing technologies have moved on since then, and we now have new technologies like this so-called long read genome sequencing. And this effectively is like having much bigger jigsaw puzzle pieces. Now, it's still not an easy jigsaw puzzle. There's still millions of these bigger pieces that need fitting together. But our team were able to piece these all together with a few other tricks as well, bioinformatic tricks, to actually assemble now the genome to the chromosome level. So now we've got 10 pieces, each one piece representing a chromosome with all of the genes on it. So to the best of our knowledge, this is the first chromosome level genome assembly for any Phytophthora species in the world. And you can see on that bottom left there the 10 chromosomes. So each of those chromosomes will contain a long DNA molecule that we now know the sequence for, and they will each have thousands of genes, and we know the genes and where they are. But we can see now why it's so very difficult to assemble these genome sequences, and that's because they have lots of repeats. So what a repeat is just a sequence that's used over and over again, the same sequence. And it's kind of like having a large number, thousands of jigsaw puzzle pieces that are completely identical to each other. And you can see those repeat regions shaded dark on those chromosomes. So if you have a lot of identical jigsaw puzzle pieces, normally you could probably put them interchangeably in your jigsaw puzzle, but here we have to make sure they go in the right place, and actually figuring out what the right place is difficult, and it makes piecing the rest of the non-repeat bits very difficult as well. So Professor Murray Cox, who you can see pictured there, he's one of our team members, and he was the very clever bioinformaticist that managed to do this very complicated jigsaw puzzle. So now we know exactly how many base pairs of DNA are in this genome, exactly how many genes across these chromosomes. So now we have the complete genetic instruction manual to the Phytophthora agathodista pathogen. So we hope that this will now be a community resource for everyone to use for many years, and also for many uses, not just for the stuff that we're doing, but for all sorts of things. So what we're doing now is trying to understand the biology of the pathogen at the molecular level in a deeper level now by looking at these genes and what they do. So clearly there's a lot of genes, so we need to narrow this down. And one of the things we're doing is to look at which of these genes are switched on or expressed in the host itself. And so we're collaborating with Rosie on that. And we hope to find out which are the most important genes, and then we can have a look at those in more detail. So ultimately, we're hoping that by doing this sort of work and understanding the biology of the pathogen in more detail, we can find some targets for disease control, or find some or develop some tools for screening for resistance in Kauri. So thank you very much to all the team. They weren't listed on the front page, but all the people listed on the left there are a really integral part of the team, including people from many different places, including USA, Oregon State University collaborators. Thank you there in the middle to our Maori partners and advisors who've been very important in this project. And also thank you to our funding providers there on the right. And thank you very much for listening and watching.
Thanks for that presentation, Rosie. If you're just joining us, we're running ahead of schedule from what might be on your program due to a late change in lineup. So we're moving to the Q&A now, as you can see, and potentially ending the session a little bit ahead of schedule also, just to inform you. So I'd now like to introduce our panel for discussion. Joining us, we have Julia, Rose, Taylor, and Julianne, and Nari. So also a reminder to our viewers that we have the live Q&A function, so please drop in your questions there. But just while that's starting to load up, we've got a question to begin with from Massey. So this is for Julia. What role do you think citizen science / community efforts can have in monitoring MR and limiting its impacts?
Thank you for this question. It's community citizen science is essential. So whenever you come across a near-term infection, please use the iNaturalis application, because this information gets feedback to us scientists, and we can know where the pathogen is in New Zealand, what species specifically it is affecting. There's still areas in New Zealand where there is no myrtle rust, so we are interested to know where these areas are. Every year there is report of new areas getting infected by myrtle rust. Every year we have report of myrtle rust on new host species as well. And scientists cannot be everywhere at the same time. We have to rely on the community to be our eyes in the forest as well.
Awesome. Got a question here from Denise for Rosie. Now that you have the genetic sequence of PA, do you also need to know the genetic sequence of coli?
No, it's really not necessary to do that. There's a lot we can find out by studying the pathogen. So our group has exclusively looked at pathogens, worked on pine diseases as well. And it can be useful to have the host genome, but it's certainly not essential. As I say, you can find out a lot of information. So just as an example, so some of the pathogen molecules that we look at are called effector molecules. And these are ones that help the pathogen to cause disease. But at the same time, the host plants often learn to recognize these and set up a defense response, sort of like their own kind of immune reaction. So we can actually, in theory, if we find the right ones, we can use these proteins, these effector proteins from the pathogen to actually screen plants, so a population of plants, and find out which ones are responding with a defense response. And by studying that sort of thing, hopefully eventually we can improve the resistance in the general population by planting out more of the ones that are showing resistance responses. So it's not necessary to know the genome in order to do that. Of the host, sorry.
Got a question here from Aaron. Is the genome complexity of PA typical of other phytophthora, or is it especially tricky beast?
Compared to other phytophthora, I think it's similar, and we're still at we're just the early stage now of starting to do comparisons with other phytophthora. But I think one of the reasons why we've been able to assemble it to chromosome level is the repeat content is a bit lower than some of the phytophthora. So some of them have far more. Some of them, ours is diploid, which means 2 copies of every chromosome. Some of them are tetraploid, or other levels of ploidy, which makes them a whole other level of complex. These are complex beasts, but there's teams all over the world working on these. And yeah, hopefully collectively we can make some breakthroughs in the next years.
And we've got a question here from Rebecca for Julianne and Nari. How important are the impacts of other Phytophthora species on coli, and what patterns are you seeing in your surveillance?
Yeah, that's a really great question, Rebecca. So one thing we do when we're doing the diagnostics for Phytophthora agathodicida is look for other pathogens, and particularly Phytophthora within the soils. And one that comes up consistently and often is Phytophthora cinnamomi. Yeah, but we are seeing that there's a fairly strong association and consistent association of Phytophthora agathodicida with the acute symptoms of coli diabetic, and the specific role of Phytophthora cinnamomi is yet to really be quantified. But there are some much earlier studies that did look into the impact of Phytophthora cinnamomi on coli, and it certainly has the potential of causing disease and killing trees under the right circumstance. So that's an area that really needs to be fleshed out. And in addition to cinnamomi, there are other Phytophthora pathogens in the mix as well. Understanding whether or not these have been introduced into the forest or whether they're actually part of the native microbial flora is something that we still also don't know, but something that we're looking into within the host pathogen environment, Mahi, as well. So thanks for a great question.
Awesome. And another question for you, Rosie, from Ariane. Will a more complete genome help to identify the origin of Phytophthora agathodicida? Sorry for my pronunciation there.
I don't think it will make that much difference to identifying origins. So that's the work mainly of Richard Winkworth has been working on that. They've used mostly mitochondrial sequences for doing that work. So it won't make a lot of difference having the complete genome. The reason why we wanted the complete genome was because a lot of the genes that are important for the pathogen to cause disease in other Phytophtheras have been found in these gene poor regions that are quite repeat rich, and those are the sort of regions that are lost in genomes that aren't assembled properly. So for example, I just had a look at the numbers the other day. We've actually got 400 more genes now than we did have in the original version. We haven't had time to look yet at what they are and what they do, but we suspect that some of those actually might be quite important for understanding the pathogen, but not necessarily for looking at the origins.
On that note, there seems to be a lot of interest in the genome sequencing work. Rosie, for myself, I'm not, a sequencer or unpacking genomes myself, but I found it quite interesting the mentioning that you had between the short read and the long read, and how there's been this progression and technology advancing, etc. Do you see anything on the horizon that might take it a step further in the near future for
absolutely, yeah. Not specifically. I mean, I'm not a bioinformaticist. People like Murray Cox do all that really clever stuff on the computer. So yeah, the technology is advancing at a rapid rate all the time. You can actually get longer reads even than what we did, but the accuracy of them is a lot lower. So we used the one that was more accurate, but also big. But Murray also used a few other tricks, like looking at where different bits of chromosomes sit relative to one another in 3D space in the nucleus, so which is quite high tech as well. But yes, this is moving on all the time, and sequencing is getting cheaper and easier. Yeah.
Fascinating. And we have another question in the chat. Just a reminder to viewers, probably last chance to add to the live Q&A if you want to have your question read. This one's from Aaron for Julia. The resistant plants you have found, how much / how long have they been exposed to MR, i.e. how tough does their resistance appear to be, and how hopeful they will be to withstand MR long term to avoid extinction?
Yes, very interesting question. Thank you, everyone. So the resistant plant that we have identified have been exposed to myrtles for the past 2 to 3 years. So these were part of a myrtles trial plantation under Narako Taketake. And it's a joint work that I'm doing with World Beresford. And we had this trial planted in Rotorua and Auckland, and in both trials, we identified what we think are resistant or tolerant Damarama. So the plants were naturally infected to myrtles. We did not perform artificial inoculation on them. So you can never be 100% sure under this condition if the plant would have escaped the disease, but we just assume that after 3 years, they would have encountered the pathogen at least more than once. We also noticed like a big difference in the growth of these individuals. They had an extensive growth. They are very tall trees, and the ones that are very susceptible to myrtles were constantly infected and ended up staying very small. So that's interesting, and there is definitely more research that needs to be done on these trees to confirm their resistance or their tolerance character. The other question, I don't I don't remember what it was. How long will they be resistant? I think that was the other part of the question. So that's a very interesting one, because at the moment we only have one strain of myrtle rust in New Zealand, and it's called the pandemic strain. So what we are observing at the moment, it's just for the pandemic strain. But there is a risk of other strains of myrtle rust to come to New Zealand, especially the strain that they have in South Africa. We have been testing under controlled condition whether this strain in South Africa could infect New Zealand species, and we demonstrated that yes, it can. So there is a real biosecurity risk that other strains of myrtle rust can come to New Zealand and overcome the resistance that we might see at the moment.
And we have one more question in the chat. This might be the final question, I think, from Nick, who congratulates everyone on a great kōrero, but has another question for Julia. Have you heard any updates of myrtle rust in your homeland of New Caledonia?
Thank you very much, Nick, for this question. I've been in New Caledonia for the past 3-4 months, and I've been doing some fieldwork for myrtle rust there. And what I can say is we are observing the same impact of myrtle rust on the native species that they have in New Caledonia, especially on Eugenia bullata and Eugenia gacognei, Eugenia brevistilis. So myrtle species that have fleshy fruits and that look very similar to Lophomertus in New Zealand. So yeah, common impact. These species have their fruits and their flowers regularly infected by myrtle rust. The people there did not see any regeneration of the population that are getting infected. So it is very likely that myrtle rust will be causing localized extinction of various myrtle in New Zealand, in New Caledonia, and also in Australia.
Fascinating. Thank you to the panel for all of your beautiful whakaaro, and thank you to the live Q&A chat for contributing to this session. I'm going to hand things back to you, Taylor, just to help wrap up the session.
Thank you very much. And yeah, massive mihi to all the speakers. That was a really, really interesting session. And I think it's always really interesting to see how that technology is changing our understanding of these pathogens, but also it's interesting to see the progression along those different journeys from, kauri dieback has been in Aotearoa, and we're learning to kind of work with that. And now we've got myrtle rust, and we might be still earlier on our journey with that pathogen. There is a slight change to proceedings from here. Our next session is starting slightly early. So our next session is actually starting at 2:05 instead of 2:15. And so we're going to end this session now. And so everyone can go have a break, have a cup of tea, and we'll see everyone back at 2:05. A reminder that you can use the chat function to chat to each other, and if you've got a bit of time to go check out those exhibitions. But otherwise, we'll see you all back here at 2:05.